Alright, so in this video we're going to go over the incremental exercise test protocol that we performed for your first lab report. So it's an incremental exercise test because every 3 minutes we go up in power output, or the amount of work your subject is doing is going to increase every 3 minutes. Okay? What we really care about is at the end of these 3-minute stages, during the final minute, we measure heart rate, blood lactate, and steady state VO2. If you're not familiar with what steady state VO2 is, you have to remember, at the start of each of these stages, VO2 is going to change very rapidly, whereas near the end of the stage, it's going to plateau and not change very much. So when you go to make the figures for your lab report, make sure you are only graphing the VO2 data from the final minute of each stage, because that represents the VO2 needed to do the workload of that stage. So here's our subject cycling. They're breathing in on the right side through that red tube, the tube with the red arrow on it. They then breathe out into the blue tube. In the middle, the mouthpiece, it has 2 one-way valves. This makes it impossible for the subject to breathe out into the red tube or breathe in from the blue tube. So all air that they breathe in comes from the red tube, and all air that they breathe out goes down the blue tube. When they exhale, all of that air goes down the blue tube to the mixing chamber. Okay? So they breathe out. All of that air, known as VE, goes down this tube into the mixing chamber, the large transparent cylinder on your screen right now, and then it's forced out the other side through this turbine. And you can see the turbine sort of flickering in the gaps there. Okay? And so this turbine tells us how much air someone is breathing out. Okay? We then have what is called a sampling line. The sampling line will tell us the fractions of oxygen and carbon dioxide in the air that someone is breathing out. And we also have a probe that tells us the temperature and the humidity of the air that the subject is expiring. That blue disc there, that's essentially, you can think of that as their mask in order to keep the tester safe during COVID-19. All of the data is then put up on a computer screen for the researcher to see. And so every minute, we're also measuring blood lactate. Okay? So how you measure blood lactate is you just saw the researcher clean the finger with an alcohol swab and dry it. He then pokes it with a lancet. He will squeeze very gently until there's a small droplet of blood. He then wipes away that drop. He does it again for good measure. In this case, he's using a capillary tube, and he has to fill that entire capillary tube with blood. Once that tube is filled, he gives the subject a piece of gauze to hold on to so that they don't bleed all over the place, and he puts that capillary tube into a smaller test tube and shakes it up. Okay? This is an example here of the researcher collecting blood lactate while the subject is cycling. So he pokes, wipes away the first drop of blood, and fills his capillary tube. And then gives subject gauze. Okay, and here's one more angle of collecting blood lactate. Remember, we also need to record RPE at the end of each 3-minute stage, and that is as simple as putting an RPE scale in front and the subject points at their RPE. So once we get the blood lactate samples, We take it, we carry it across the room, in this case anyway, and we plop it into the blood lactate analyzer. Okay? The analyzer takes small little samples from each of these tubes, and then it will display for you what the subject's blood lactate and blood glucose levels were.